Hi, everyone. Welcome back to The Bewitching Brain. My name is Kaelin Sagraco, and we are so happy to have you with us and encourage you to subscribe and share. Take good care, be safe, and help others be safe. We hope you have been enjoying our series, helping you make sense of your sensational senses. So far, we have touched on your sense of touch, vibration, pain, smell, and taste. So if you're interested in any of those, please look back at our previous videos on our page. For this week's video, we are taking on a completely different sense, one that manipulates the type of energy present, from mechanical to electrical to chemical, your sense of hearing. This sense has a lot more steps to it than the previous ones, so if you're interested in this complex transduction pathway, stick around for more bewitching brain content. Once again, I will sound like a broken record and start with the receptors responsible for auditory transduction. In your sense of hearing, you have two different types. These are your inner and outer hair cells. They are called hair cells because they have what are called stereocilia that are connected to mechanically gated receptors. They work it like knocking over dominoes. Once one is displaced, it will knock the next one down, stretching the receptor membrane, allowing ions to rush into the cell. These different types of hair cells serve different purposes as well. As for your inner hair cells, these are responsible for what you imagine auditory receptors would do. They send afferent sensory signals to your brain about what you are hearing based on the frequency of the displacement of your stereocilia due to a sound wave. As for your outer hair cells, these are ones responsible for protecting your inner ear by efferently or back from your brain, amplifying or dampening incoming sound information. Think about going to a really loud rock concert. Your inner hair cells will tell you what song is playing, while your outer hair cells will recognize that, wow, this is some really loud music, and dampen the incoming sound waves to protect your ears from any supposed damage. Cool. So now we know that how these cells are getting and processing the information from sound. But where are these receptors located, and how do they eventually get activated? Well, it starts when the sound wave travels through the air into your ears. Once it enters here, it will vibrate what is called your tympanic membrane, or your eardrum. These vibrations will cause what is known as your ossicle bone chain to hit your cochlea at appropriate frequencies. Your cochlea is composed of 3 canals filled with fluid. These canals are the tympanic, vestibular, and middle canals. Another important membrane is the one in between the middle and tympanic canals, known as the basilar membrane. The basilar membrane holds all of your hair cells. Once the sound waves are transduced through the cochlear fluid, it will displace this membrane and displace the stereocilia as mentioned before. Once these stereocilia are displaced and our auditory receptors are activated, this information gets transduced along a cranial nerve to your auditory cortex. This auditory cortex is located in your temporal lobe, which is conveniently located right next to your ears. The specific cranial nerve, as mentioned before, in the different types of hair cells, has both afferent and efferent projections. This means that your auditory cortex both receives sensory information from your inner hair cells and also projects efferently to tell the outer hair cells how to amplify or dampen the sound being processed. The last thing we should go over about auditory processing is pitch and location determination. When it comes to determining what pitch or tone you are hearing, we use 2 different types of coding. These coding systems are place and temporal coding. Place coding deals specifically with the basilar membrane, the membrane that holds all of your hair cells on it. The sound waves will vibrate the basilar membrane at different locations depending on the pitch you are hearing. High frequencies or high-pitched sounds will vibrate the base of your basilar membrane, whereas low frequencies will vibrate the end or apex of your membrane. The other way is through temporal coding. Temporal coding refers to the frequency of action potentials your receptor cells are experiencing, relating or coding the pitch you are hearing. This means if your inner hair cells are sending action potentials at a very fast rate, your brain perceives this as a high frequency and high pitched tone, and vice versa. Last but not least, determining the location of the sound you're hearing. Being able to process that you are hearing a hive of angry bees is very important, but equally as important is to know where that sound is coming from so you can run away. This can be processed by what are known as binaural cues. The binaural cues include intensity and latency differences. Intensity differences are simply being able to process the difference of loudness between the two ears. If your right ear is processing a louder sound from the beehive, it is most likely on the right side of your body. The other way we track sound is latency differences. This refers to the difference between the time of sound wave arrival. Though very close in timing, your brain is able to recognize that your right ear is being activated before your left, meaning that what you are hearing is to your right. Thank you so much for tuning into this installment of Bewitching Brain. Tune back in next time for an overview of your sense of sight. Thanks for listening, Brainiacs. Can't wait to see you again.